قبل الصلاة. اوكي، بسم الله الرحمن الرحيم. احنا هنتكلم عن physiology of the lower GIT. هنبتدي بالسمول intestine motility. Small intestinal motility is regulated by myogenic, neural and hormonal factors. الميوجينيك فاكتورز هي الأهم على الإطلاق. نيورال وهرمونال فاكتورز بتساعد to modify الميوجينيك اكتيفيتي. حتى لو حصل بلوكج للنيورال اكتيفيتي هتفضل الانتستين شغاله بالميوجينيك اكتيفيتي الانتريك. There are two phases of motor activity. In the fed state, the food is moved through the intestine by segmentation and peristalsis, while in the fasting state, the motor activity continues through the migratory motor complex. The migratory motor complex moves undigested material into the colon and prevents bacterial reflux from the colon into the ileum. Motilin and ghrelin are believed to initiate the migratory motor complex, while cholecystokinin and gastrin acting to oppose it. It originates in the stomach and progresses distally through 4 phases. Phase 1, no motor activity. Phase 2, disorganized accelerating high pressure waves. Phase 3, continuous rhythmic contractions. And phase 4, the contractions once again become intermittent. This cycle repeats every 1.5 to 2 hours. Autonomic nervous system control of motility. The sympathetic control affects the enteric nervous system and directly innervates smooth muscles, endocrine, and secretory cells, inhibiting digestive and secretory functions. Sympathetic stimulation primarily reduces propulsive force, with alpha adrenoreceptors playing a key role in inhibiting intestinal motility. Neostigmine promotes motility by inhibiting acetylcholinesterase at the neuromuscular junction, while metoclopramide increases upper GI motility by enhancing tissue sensitivity to acetylcholine. Parasympathetic innervation mediated by preganglionic nerve fibers that synapse on the enteric nervous system neurons, which can be cholinergic or peptidergic. Enteric nervous system control of small intestinal motility. The enteric nervous system independently regulates motility and facilitates the execution of sympathetic and parasympathetic signals. The ENS receives sensory input from mechanoreceptors and the chemoreceptor in the bowel wall, responding by directly innervating smooth muscle, secretory, and endocrine cells. Segmentation involves neural inhibition and disinhibition of adjacent bowel segments, likely pre-programmed into the ENS with burst-type activity as the pacemaker. Opiates cause a non-propulsive, constipating segmentation pattern. Peristaltic reflex is triggered by distension. It propels chyme along the small intestine through reciprocal muscle actions, longitudinal muscle relaxation behind the bolus and contraction ahead, while circular muscle contraction behind the bolus and relaxation ahead. The peristaltic reflex is mediated by acetylcholine, substance P, VIP, serotonin, and nitric oxide. Ileal brake reflex. This is triggered by partially digested triglycerides. It inhibits jejunal motility, increasing transit time for nutrient absorption, potentially mediated by peptide YY, which is the neuropeptide, like that. Hormonal control of small intestinal motility. GI hormones and intrinsic pacemaker. Hormones like gastrin and cholecystokinin, secretin, and motilin rapidly peak within 20 to 40 minutes, followed by a decline, and modulate the intrinsic pacemaker. Gastrin increases small intestinal contractions. Impaired peak levels of gastrin are seen in patients with constipation predominant functional bowel diseases. Serotonin is stored and secreted in large amounts by small intestine. It promotes intestinal motility and secretion, playing a key role in digestion. So, serotonin modulation using SSRIs has been effective in treating constipation predominant IBS. This is a summary of the pro-motility hormones and anti-motility hormones. Pro-motility hormones include gastrin, cholecystokinin, motilin, VIP, while anti-motility include secretin and glucagon. Small intestinal secretion includes mucin, bicarb, and water. The mucin is produced by goblet cells, essential for innate immunity in the gastrointestinal tract, has protective effects In the stomach and the duodenum, the mucin layer forms a protective pH gradient and acts as an antimicrobial barrier. Mucin glycoproteins are toxic to bacteria and anchor IgA and antimicrobial peptides. Secretion of bicarb. It's secreted by duodenum. It neutralizes gastric acid and protects the mucosa, with the duodenum secreting the highest bicarb rate. Bicarb secretion is driven by cystic fibrosis transmembrane conductance regulator and requires energy and carbonic anhydrase. Secretion of water in the small intestine to maintain balance between absorption and secretion. There is passive water movement. Water moves passively across osmotic gradients, either paracellular or transcellular via aquaporins. Secretion is regulated in the small intestine by the enteric nervous system and hormonal regulation. Those are the hormones regulating secretion in the small intestine, with prosecretory hormones including serotonin, VIP, cholerotoxin, and substance P, while anti-secretory like neuropeptide Y, which is peptide YY, and anti-secretory factor. Small intestinal mucosal immunity. The small intestine serves as the largest immune barrier between the body and external agents, interacting with foreign proteins, viruses, bacteria, and toxins. The mucosal immune system, comprising mucosa-associated lymphoid tissue, accounts for approximately 80% of the body's immune cells, with 70% located in the gut-associated lymphoid tissues. Three main functions of the mucosal immune system in small intestine include preventing harmful microbial colonization and invasion, blocks undigested foreign antigens from food and commensal bacteria, prevents immune responses that could harm the host. Mucosal surfaces are constantly exposed to foreign material, requiring careful regulation of immune responses to prevent self-harm. The system includes both innate and adaptive immunity components. The immunity is mediated by Paneth cells, which contribute to innate immunity by exocytosing bactericidal granules in response to inflammation. Paneth cells contain antimicrobial proteins including lysozyme, alpha defensins, HD5 and HD6, phospholipases. A reduction in HD5 or HD6 is associated with ileal Crohn's disease, while secretory phospholipase A2 is released by Paneth cells. This aids in immune response and tumor suppression. Paneth cells express tumor necrosis factor alpha, which is implicated in crypt regeneration. Paneth cells can express NOD2, a gene linked to Crohn's disease susceptibility. Immunity cells include microfold cells, or M cells, which are specialized cells critical for mucosal defense as they sample pathogens and foreign antigens from the intestinal lumen. M cells have a concave basolateral surface which facilitates interaction with antigen-presenting cells. M cells function in transcellular transport, internalizing particles, including microorganisms, with minimal processing. Intestinal macrophages are essential to the immune response. It functions by playing a key role in clearing bacteria. They help maintain immune balance and protective immunity. Intestinal macrophages are highly phagocytic and bactericidal, clearing bacteria without triggering inflammation. These macrophages do not release pro-inflammatory cytokines or express co-stimulatory molecules. They secrete anti-inflammatory cytokine interleukin-10, with loss of interleukin-10 leading to spontaneous colitis in mice. They produce papaver gamma to suppress pro-inflammatory gene expression. Dendritic cells act as the antigen-presenting cells in the small intestine. They are located in the Peyer's patches and isolated lymphoid follicles and mesenteric lymph nodes. T cells in the small intestine interact with the dendritic cells. There are two main populations of T cells: intraepithelial lymphocytes, which are predominantly CD8 positive, and lamina propria lymphocytes, with equal distribution of CD4 and CD8 cells. IgA in the small intestine, it is the most abundant immunoglobulin in the body, with the gut producing 80% of the total IgA. There are two forms of IgA, IgA1 in small intestine and IgA2 in the colon. Okay, small intestinal digestion and absorption. The primary function of the small intestine is absorbing nutrients, water, and vitamins from the food. Protein and carbohydrate digestion begins before food reaches the duodenum. The small intestine entirely responsible for fat digestion. The entire process of digestion and absorption typically takes 3 to 6 hours. Carbohydrate digestion, including starch digestion by pancreatic amylase, then monosaccharidases in the brush border enzymes. Protein digestion starts in the stomach with pepsin and continues in small intestine with help of pancreatic enzymes. Pancreatic proteases are secreted as inactive proenzymes and activated in the duodenum by brush border enzymes. Activation of pancreatic proteases, the main proenzyme involved is trypsinogen, which is activated to trypsin by enterokinase, which is a brush border enzyme. Then trypsin activates other proenzymes like chymotrypsinogen, proelastase, and carboxypeptidase. These enzymes break down proteins into oligopeptides and amino acids, which are further hydrolyzed by brush border peptidases. Free amino acids, dipeptides, and tripeptides can be absorbed by enterocytes, while dipeptides and tripeptides are absorbed more easily via a proton gradient. Amino acids are absorbed via sodium-dependent active transport. Once inside the enterocytes, dipeptides and tripeptides are converted into amino acids by cytosolic peptidases. Amino acids, particularly glutamine, may be used for energy or protein synthesis in the enterocytes. Other amino acids enter the portal circulation. Fat digestion. Fats are ingested as triglycerides, phospholipids, cholesterol, and cholesterol esters, with triglycerides comprising 90% of dietary fat. Fat digestion begins with bile salts from the liver, which emulsify large fat globules, breaking them into smaller molecules accessible to enzymes. Pancreatic lipase breaks down triglycerides into free fatty acids and monoglycerides. The components are absorbed into micelles composed of bile salts and lecithin, facilitating transport to the brush border. At the brush border, the micelles disintegrate, allowing fatty acids and monoglycerides to enter the enterocytes. Inside enterocytes, the fatty acids and monoglycerides are transported to the smooth endoplasmic reticulum, where triglycerides reform. They are packaged into chylomicrons in the Golgi apparatus. Then chylomicrons and apoproteins are released via exocytosis into the lacteals entering the lymphatic system. Short and medium fatty acids are absorbed directly into the portal blood. Most fat is transported as chylomicrons to the thoracic duct. Cholesterol is absorbed as VLDL, which also enters the lymphatic system. Most fat absorption occurs in the duodenum and proximal jejunum. Okay. The enterohepatic circulation includes the bile salt pool in humans averages 2 to 3 grams, with about 95% being reabsorbed in the portal circulation. They are absorbed either passively or actively. Bile salts are resecreted and stored in the gallbladder, which is the enterohepatic circulation. This occurs approximately 6 times a day. Patients with ileal resection lose the ability to reabsorb conjugated bile salts, leading to excess bile salts in the colon, which can cause diarrhea due to impaired sodium and water reabsorption. This can be treated by cholestyramine, which is a bile acid binding resin. Digestion absorption of vitamins, including fat-soluble vitamins, vitamin A, D, E, and K, they are incorporated into micelles, taken up by enterocytes, then by chylomicrons into the lymphatic system. Water-soluble vitamins are absorbed in the jejunum and ileum via active or passive transport. Vitamin B12 absorption occurs through binding to intrinsic factors secreted by gastric parietal cells. The complex of vitamin B12 and intrinsic factor binds to a receptor in the terminal ileum for absorption. After entering the enterocyte, vitamin B dissociates from the complex and enters the portal circulation. Conditions affecting intrinsic factor availability or the ileal receptor, such as gastrectomy, gastric bypass, or ileal resection, can cause vitamin B12 deficiency. Water and electrolyte absorption. The small intestine processes 8 to 10 liters of fluid daily from dietary intake and various secretions. Water absorption occurs via passive diffusion or osmotic pressure gradients driven by electrolyte absorption. The proximal small intestine, water enters freely throughout tight junctions. In distal intestine, it is absorbed through active transport coupled with electrolytes. Sodium is absorbed via active transport. Bicarb absorption involves proton secretion forming carbon dioxide and water. Calcium absorption in the duodenum and jejunum is regulated by parathyroid hormone and vitamin D. The active form of vitamin D enhances calcium binding proteins in the enterocytes. Nearly 35% of dietary calcium is absorbed. Iron absorption in the duodenum occurs in the presence of bile. Apotransferrin binds to iron and facilitates its uptake into the enterocytes. دكتور محمد احنا هنعمل في التايم مانجمنت ايه؟ يعني احنا فاضل لنا
الكولون we will cover it in the next wave مش
Okay.
مش في المرة دي.
Okay.
If we finish the small intestine,
Okay.
thank you very much.